Okay, good morning or good afternoon, and in this lecture, we will be covering drug and chemical standards for pharmacy. So, as future pharmacists, you might want you might fall into different fields that would necessitate knowledge on drug and chemical standards. Take for example, you're going to be a compounding pharmacist and you have to decide on what certain types of ingredients would best suit your preparations. And if you are a quality control specialist for a drug manufacturing company and you have to reference different grades of chemicals, this is the lecture appropriate for those skills to be developed. So, being as a pharmacist in the future, you should be knowledgeable in the different drug and chemical standards used in our pharmacy practice. Okay, so to start, let us first discuss the history and the changes that happened to our United States Pharmacopoeia and the National Formulary. So our USBNF are the most prominent drug reference standards for the whole world for that matter. Here in the Philippines, this is the go-to reference for drug companies and for our compounding pharmacists. So historically, Pharmacopoeia has its origins on its Greek meanings, which is pharmakon, which means drug, and poen, meaning make. So when you combine the two words together, it means how to make a drug. So a pharmacopoeia is actually a way or a recipe or formula required to make or prepare a drug. However, in the modern times, our pharmacopoeia actually means a uniform set of national drug standards. So when we say pharmacopoeia, it's basically a reference on official preparations that we can check our preparations with. So a pharmacopoeia is basically a reference book. So in the US, drug standards were provided on a national basis in 1820 when the first USP was published. But before the USP was published, the very first American pharmacopoeia was the Lititz Pharmacopoeia. It was published at Lititz, Pennsylvania, for use by the military hospital for the US Army. It contained information on 84 internal and external drug preparations. Now, our Lititz Pharmacopoeia is not a national pharmacopoeia, or nor is it an official pharmacopoeia. It is just the very first pharmacopoeia to be recognized. The first pharmacopoeia to be the first national pharmacopoeia is actually the United States Pharmacopoeia. So on January 6, 1817, Lehman Spalding, a physician from New York, submitted a plan to the Medical Society of the County of New York for the creation of a national pharmacopoeia. Spalding's effort were later to result in his being recognized as the father of the United States Pharmacopoeia. So why was there a need for a nationwide pharmacopoeia? So the main problem they encountered was if there are different standards for drugs and drug preparations, if you take your prescription out of state or if you move from one state or to the other and you brought your prescription with you, the next pharmacist might have a chance that he does not recognize the drug preparation in your prescription, nor can he recognize what are the active ingredients or what even the prescription contains. So this problem is exacerbated over many different states in which they have different drug standards. They might even have different names for different chemicals or active ingredients. So to remediate this, to solve this problem, they developed a national standard. Okay? So the national standard was our United States Pharmacopoeia. So draft pharmacopoeias were submitted to the convention. These were reviewed, consolidated, and adopted by the first United States Pharmacopoeial Convention assembled in Washington, D.C. So only the United States Pharmacopoeial Convention can make a new volume of our United States Pharmacopoeia. So you may think of the United States Pharmacopoeial Convention as a meeting of physicians and pharmacists to decide on matters relating to official drug preparations. So the first USP was published on December 15, 1820 in both English and Latin. Why did they choose Latin? It's because Latin was a primary language used in the medical sciences at that time. So what was the main purpose of the USP? So this is the objective of the first USP. It is the object of a pharmacopoeia to select from among substances which possess medicinal power, the utility of which is most fully established and understood, and to form from them preparations and compositions in which their powers may be exerted to the greatest advantage. It should likewise distinguish those articles by convenient and definite names, such as made to prevent trouble or uncertainty in the intercourse of physicians and apothecaries. So you can divide the two. You can divide the first objective into two main parts. So the first objective of a pharmacopoeia is to determine which substances have medicinal abilities. Second is to form preparations or formulas from these different medicinal agents. And it also should distinguish those articles by having a definite and common name. So the problem of having different common names for the same chemical substance is solved by having a official name for that substance. So if that drug substance is called three names by different counties or states, in the USP it is only called by its official name, meaning all counties or states would recognize the official name, leaving out any room for confusion. So the revisions of the USP. So when was the USP revised? So the first provisions in the very first pharmacopoeias stated that every 10 years there would be a new USP. However, it also granted authority to issue supplements to the USP whenever it is necessary to maintain satisfactory standards. Now, because of rapid development in drug discovery, there were more and more drugs that necessitated to be added into the USP. So as such, in 1940, in that year of meeting, it was decided that the USP would be updated every 5 years while still being allowed to use periodic supplements to add knowledge to the USP in between publications. So where does the National Formulary belong? So when the American Pharmaceutical Association was established in 1852, the only authoritative and recognized book of drug standards was the USP. So to serve as a therapeutic guide to the medical profession, its scope then and now was restricted to drugs of established therapeutic merit. So because of the strict selectivity of the USP, many drugs and formulas that were not accepted but are used by the medical profession were not granted admission into the early revisions of the USP. So what happened is that there were preparations being used in the market that were not official. So there are unofficial preparations. And the problem is, if it is unofficial, it's not in the USP, not everyone knows how to prepare that formulation. So as a type of protest, and while keeping with the original objectives of the American Pharmaceutical Association to standardize drugs and formulas, certain pharmacists prepared a formulary containing many of the popular drugs and formulas denied admission to the USP. So they made their own pharmacopoeia. So the first edition was published under the titles National Formula of Unofficial Preparations. So this was the original name of our national formulary. Now, the title changed to National Formulary in June 30, 1906, when President Theodore Roosevelt signed into law the first Federal Pure Food and Drug Act, designated both USP and NF as establishing legal standards for medicinal and pharmaceutical substances. So what this led to was there were two official compendia. Both the USP and both our NF are recognized as official compendia or official drug standards. So the law required whenever the designation USP or NF was used in labeling, it must conform to the physical and chemical standards set forth by the monograph. So if you're ever going to encounter drug labels that contain paracetamol USP, not paracetamol alone, it is paracetamol, it's called comma USP. This means that the paracetamol used in that drug preparation is compliant to the minimum standards set by the USP monograph. Only drug substances that are fully compliant with the monograph in the USP or NF can use the suffix USP or NF in the ingredient listing. So the early revisions or early editions of the National Formulary served mainly as a convenience to practicing pharmacists by providing uniform names of drugs and preparations and working directions for small-scale manufacture of popular pharmaceutical preparations. So in the modern NF, our NF contains various pharmaceutical preparations. So these are inactive substances but are primarily used in compounding. So you will see our ointments, our creams, our topical preparations in our NF. You can also see our suppositories in our NF. So as you can see on the screen, you can see the monograph of our epinephrine. When we say a monograph, it is basically a characteristic page of a certain drug. If you want your drug to be labeled as USP grade, your drug sample must be able to pass all the tests stated in the monograph, including purity, absence of contaminants, and the monograph also contains methods and testing on how to identify your drug if ever you would need them. Okay. Now, if ever you're going to encounter USP grade chemicals, that simply means that it is compliant to the minimum standards set by the monograph. USP grade chemicals are usually more expensive. So having two formularies were actually a little bit inconvenient since you had to refer to two different books. So in 1975, the United States Pharmacopoeial Convention Incorporated, so this is the company that handles the USP, purchased the National Formulary, thus unifying official compendia and providing the mechanism for a single national compendium. So in 1975, it was combined, and in the first national compendium, which composed of the USP and NF together in one package, became official on July 1, 1980. So from then on, USP and NF were always together. Okay? It started on 20 for the USP and 15 for the national formulary. Please take note that our USP and NF will never have the same volume number in the same time. It's because USP was ahead of NF by 5 years or by 5 volumes. So what is the current USP now? The most current USP is our USP 43 and NF 38. So as you can see on the slides, this is the physical copy of our USBNF. These are various, usually USB outnumbers NF by a ratio of 5 books to 1. And NF is slightly less thick than our USB. And you can see the pricing of the USB and NF here on the screen. So just so you know, USBNF is not being published as textbooks anymore. It's because the publication of a physical copy actually increases the cost for the end user. So instead, USBNF has stopped publishing actual books and references and instead moved on to an official database. It's like a online subscription, so you can access the latest USB and NF. Now, as you can see on the slides, the price is actually around $700 or around 34,500 pesos. It's a little bit expensive, but it's not actually for regular pharmacists. So if you're a community pharmacist or a teacher like me, you don't actually need a copy of the USBNF. So who are the persons who need a copy of the USBNF? So if you're a compounding pharmacist, it might be useful to have a subscription on hand if ever you're going to check the quality and purity of the ingredients you will be using to make new drugs. And if you're a quality control specialist for a drug company, it might also be useful, and it will always be useful if ever you're going to check your preparations against a certain standard, or if you're going to check if your drug or raw material is actually compliant to our USP and NF grade materials. So here are some general chapters for our USP and NF. So you may want to read them, but these are mostly important for our practicing pharmacists. So if you're ever in the field of non-sterile compounding, you are going to read 795. If you're in the compounding of sterile preparations such as eye drops, IV solutions, anti-cancer drugs, you will want to read 797. If you're handling dangerous drugs such as those that can cause abortion or those that can cause cancer, please read chapter 800. And if you're going to be in charge of overall compounding processes, read 1163, which is for quality assurance. The 1160 is particularly, you don't actually need to read this since you have passed pharmaceutical calculations, but if you're going to review pharmaceutical calculations, it might be lenient for you to read 1160 as well. So other pharmacopoeias, so these are not only USP and B and NF are the only pharmacopoeias in the world. There are other pharmacopoeias and there are other regulatory agencies that will recognize different pharmacopoeias, but the most prominent still is our USP and our NF. So you have Homeopathic Pharmacopoeia of United States, the International Pharmacopoeia, the European Pharmacopoeia, the British Pharmacopoeia, the Japanese Pharmacopoeia, and the Philippine Pharmacopoeia. So let's move on to our ingredient standards. So throughout history, pharmacists have used chemicals and other materials for prescription compounding. In the past, these chemicals and materials were obtained from natural preparations, raw materials, and household ingredients. Today, compounding pharmacists use chemicals from various sources depending on availability. So, if you're going to be a compounding pharmacist, you will have different sources of chemicals. And you should be knowledgeable on the different standards and grades of our chemicals. So, as a pharmacist, you are responsible for compounding preparations of acceptable strength, quality, and purity with appropriate packaging and labeling in accordance with good pharmacy practices, official standards, and relevant scientific data and information. So, the compounding of quality preparations must involve the use of high quality materials. If USP and NF grade materials are used to prepare compounded formulations, the preparation must meet the requirements of continental monographs. So here are some descriptions of our chemical grades. So when we say USP or NF, it simply means that your chemical meets the minimum standards set by the USBNF for that particular chemical. So it meets the minimum active ingredient present. It meets the absence of contaminants that may be toxic, and it also meets the physical characteristics. So if your chemical is USP-NF grade, you are rest assured that this chemical is safe for human consumption. When we say ACS grade or ACS reagent, this means high purity and it conforms to minimum specifications set by the Regent of Chemicals Committee of our American Chemical Society. So ACS reagent is mostly used for general chemicals. If it is ACS grade, it simply means that it meets the ACS guidelines or the ACS standards for that chemical. This is mostly used for scientific use or experimental use. Next is chemically pure. When we say chemically pure, it is more refined than technical or commercial grade, but of still unknown quality. So this is mostly for when making non-drug products. So take for example, you're going to use soap, or you're going to make soap. You should use ideally chemically pure lye or sodium hydroxide if you're going to make hard soap. The purity is more pure than technical grade, but it's not free from contaminants. It's because the use of these chemicals are just to manufacture. It's not as critical as testing or quality control. So it's okay that these are slightly contaminated since the contamination will not affect the final product. When we say technical or commercial grade, these are indeterminate quality. These are usually used for their chemical applications. So a good example of this is muriatic acid. So if you're going to use muriatic acid to clean tiles in the bathroom, you do not need chemically pure hydrochloric acid, nor should you need ACS reagent grade hydrochloric acid. That would be very expensive for that use. So we use technical commercial quality if we're going to simply use it for its chemical purpose, like cleaning or disinfecting, or use it for other other more practical uses. Okay, so technical or commercial quality are relatively cheap in nature. That is why we can use them as cleaning materials. When we say analytical reagent, these are high purity, low contaminant grade reagents used in quantitative analysis. So these are used for quality control. If you're going to perform quantitative tests or qualitative tests, you are to use analytical grade reagents because they have low contaminants that can affect the results of your analysis. Next, you have your FCC grade or your Food Chemical Codex grades. So this conforms to the standards set by the FCC. So if you're going to use these chemicals, if these chemicals are FCC grade, you are rest assured that these are safe for food use. So if you're going to manufacture food, FCC grade chemicals are usually for preservatives, colorants, and surfactants. Next is your HPLC grade. So HPLC grade is reagents used to be used for HPLC experiments or our high performance liquid chromatography. So these are basically analytical reagents with extra processing. The difference between HPLC grade and our analytical reagent is that HPLC grades are particularly absent of dissolved gases. There is no bubbles nor are there any presence of dissolved gases because these chemicals will undergo compression. If there are gases within the liquid, it would actually form bubbles, and if it forms bubbles, it would form gaps, and it would affect the spectrogram reading in the analyzer of our HPLC. That is why HPLC grade is expensive because of the additional step of degassing and removal of bubbles. Spectroscopic grade, these are chemicals used in particular for spectroscopy. It might be FTIR, UV-Vis, GCMS, or flame spectrophotometry. Primary standards, on the other hand, these are standards that only serve a purpose to be referenced. So if you buy primary standards, these are not used for reactions. These are simply used as a checking material. So if ever you're going to produce a chemical and you want it to be near a primary standard, you will use primary standards as your reference material. If you're going to compare physical properties, you get a sample of your product, you get a sample of the primary standard, you compare them. If they match, good. If they don't match, you have some explaining to do. And finally, you have a reference library for compounding for non-sterile preparations. If ever you're going to be in the pharmacy and you need to compound certain preparations and you have to review how to compound them, these are books that are available that give you general guidelines on compounding non-sterile preparations. Okay? So if you are a business owner or a pharmacy owner, you should have at least Remington's Art and Science of Pharmacy, since this is a mandatory requirement for opening a drugstore. You might also have Ansel's pharmaceutical dosage forms from our current course, and you might also have pharmaceutical calculations from our PharCal, which is your PHAR 2. Okay? And finally, before we end, I would like to show you some samples of the different grades of chemicals. Okay, so if you see on the screen, this is our Sigma Aldrich website. So our Sigma Aldrich is a chemical company in which if you're ever going to need certain chemicals, this is a good reference point since it gives you live pricing for different chemicals. So what you see on the screen is nicardipine. So nicardipine is a cardiovascular drug used for the treatment of stroke. Now, as you can see in the product part of our screen, there are several types of nicardipine that you can buy. So there is nicardipine hydrochloride powder greater than 98%. You have nicardipine hydrochloride, United States Pharmacopoeia reference standard. You also have different standards present. But let us take a look at the pricing difference between our 98% and our USP reference grade. Now, check that 1 gram of our 98% is 243 Singapore dollars. Well, 200 milligrams of our USP reference standard is 676 Singapore dollars. So there is a huge difference in pricing. The difference in pricing is actually because these two nicardipine hydrochloride has actually different uses. So the first one, nicardipine hydrochloride powder 98%, this might be used in drug formulation or drug preparations. So you can buy nicardipine in these amounts and you can compound it to other medicine. Does this mean that this is USP grade? No, it does not mean this is USP grade. However, what you can do is buy it, then manually check it if it meets the USBNF criteria. If it meets the USBNF criteria, good, you can use it as an USBNF grade material. If it does not meet the USBNF, simply you can still use it, but you cannot declare it as USP or NF. Simply declare nicardipine hydrochloride. Now, what about this one on the bottom? So this is the reference standard. When we say reference standard, it is a perfect sample based on the USBNF. So what is written on the USBNF is this sample. This sample is a perfect sample in which it is a representation of our reference. Meaning it fully complies. There is no question about it that this substance fully complies with our USBNF. So does it mean this is the best drug to be used in compounding? No, actually. It's very expensive and it's not practical for you to use it in compounding. So what is the use of this compound? If you're going to use the reference standard, there are a few reasons. You're in a quality control laboratory and you want to verify physical and chemical characteristics of your raw materials or your raw material drug compared to your reference standard. So if you want to compare properties from the standard to your purchased drug, you can do so. Or if you want to validate some testing procedures using the reference standard, which is perfect, you can use it to validate tests. So reference standards have a use, but it's just not in drug preparation. This may be used in quality control or validations or simply as a backup reference in any case that you want to double check your testing procedures. Okay, so the main takeaway here is purchase a chemical grade that would suit your needs. If you're going to make a drug, use the quality that is appropriate for drug manufacture. If you're going to validate your controls, buy a reference standard since it is a perfect reference. Okay, so that just goes to show the amount of nuance that goes into preparing different chemicals. So that would conclude my lecture for today. I hope you have learned from our short lecture. And if you have any questions and clarifications, you may ask me in our next meeting or you may leave it down in the comment section. Okay, so that would be all. Thank you for listening and I hope you have a pleasant day ahead. Thank you.